Okay, welcome to week 7. In today's last week we discussed about basic Linux commands and how to use these commands to analyze genomic data, primarily genomic data. But yeah, so uh, today's class will be talking about overviews of various not genomics, multi-omics projects. So let's get started. Okay. So first is the Human Genome Project. So we all know about Human Genome Project. So this started around 1980s. The idea was to map out the major parts of the genome because some part of the genome was too complicated back then. The segmental duplicated parts or the telomeric or the centromeric parts, those were tricky areas to sequence. So they targeted the major sections of the human genome which had protein coding genes and other non-genic intronic regions. So this was a international collaboration started in 1980, and it took around 23 years to get completed, right? So yeah, so it was coordinated by the NIH, the National Institutes of Health, and some other countries like Japan, China, Germany, etc., and UK. They contributed to the project, and it took it started. The plan was uh, drafted in 1980s, and the project started around 1990, and the deadline was 15 years, but it took around 13 years to complete, 2 years ahead of its schedule. So during the course of the project, new sequencing technologies came along, and that helped actually fast track the process. But yeah, so the goal was to provide a complete and accurate sequence of 3 billion base pairs that make up the human genome and find all estimated like 20,000 to 25,000 human genes. So we know there are like more genes now. So there are some non-coding genes which codes for non-coding RNAs, which actually do not code for proteins, but those genes are also important because these RNAs actually help regulate different gene expression, other protein coding gene expression in the cell. So yeah, so the target was to do all that, and also to sequence genomes of some other important model organisms like the mouse, fruit fly, rats, etc., the model organisms that we generally conduct experiments on. uh, but yeah, so yeah, so this was another important part, which was the ethical, legal, and social implications. So there was a committee and a association set up for this, for genomic research. So there are a lot of ethics involved in genomic research, specifically human genome, because the genome actually tells everything about an organism. So the genomic data is also needs to be handled very carefully and with proper ethical and privacy measures. So yeah, that was also set up then. So what did it do? So it generated the basically almost like 98 to 99% of the entire human genome. The rest of the genome was too complicated to get resolved at that time, as I was talking about the telomeric regions, the duplicated regions, and the centromeric regions, etc., etc. Also, it sequenced other organisms like yeast, roundworm, fruit fly, etc. Uh, and also, this data was published. The publication was published in the Science Journal, and it gave free access to the reference of the human genome, which is the first reference of the human genome, so that people could use it in the research. And the idea was like the most important part of getting a human genome sequence is to uh, for other future projects was to kind of have a reference genome so that you can do your alignment to the reference genome for future projects and so on. So without this particular project, no genomics research would have been possible, right? So again, it kind of gave a detailed information about sequence, structure, and function of a complete set of human genes. So these were basically uh, the, the inheritable instructions of developmental and functional consequences of the human genome, right? So we after that we kind of looked into proteins, the genes, uh, the proteins the genes coded for, and also helped to build certain databases for all the knowledge. So yeah, this was the project that allowed the blueprint for building a human genome, right? So and again, due to the human genome project, there was a lot of boom in the sequencing technology industry as well. So a lot of new technologies came out. I think 454 Roche came out around the time of the completion of the human genome project. So we were moving towards the second generation of sequencing technologies, and that also kind of brought down the cost of sequencing a genome almost 10 to 20 times. So that was also a very important part. So yeah, it 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 helped support researchers who actually were finding homologous genes with different functions. So the sequences of the genes were very similar, but we found different functions of the genes. So those were also very important in back in the day, and also differences and similarities of these sequences with other organisms, including mouse and fruit fly, etc. So we kind of could compare our genome, the human genome, with the genome of the model organisms that we use for the experimental purposes. So that was very important back then. And we actually, due to the sequence similarity and differences, we could discover functions of particular genes and identify genes which were critical to life. So that, yeah, very impactful. So this is kind of a timeline of the Human Genome Project. It started around 1990. Then the first human genome centers, US genome centers, were established around 1991. Then the second generation genetic map was developed, and then we had a 5-year plan for the Human Genome Project, came out in 1993. First mapping goal was achieved in 1994. Then the first human gene map was established in 1996, and the first archaeal genome was also sequenced at that point of time, and also the East genome was also sequenced. And then the US uh, generated an institute called the NHGRI, which is the National Human Genome Research Institute, which kind of handled these different projects, and scientists who were interested in doing genomic research became associated with it. And after that, the first full scale human genome sequencing began in around 1999, and it got completed around 2000 and 2001. And the first paper was published in Science, and then also Nature. And this was the first draft of the human genome that was published ever. So yeah, so we also got like 10,000 full length human cDNAs, also complementary DNAs, which are basically RNA converted to DNAs. So we could sequence a few RNAs at that point of time as well. And around 2002, the first version of of the mouse genome was also sequenced and published. And 2003 kind of marks the end of the first version of the human genome project, right? So it took like 13 years. The goal was 15 years, but due to advances in technology at point of time, the the goal was achieved faster, and also the cost of sequencing a human genome came down drastically. Okay. Okay, so now after the first human genome project, then we looked into, so we found different variants in the genome. So after the first human genome project was established, we had the full reference sequence of the human genome. And after sequencing multiple other human beings, we found that there are variants in the genome, right? So that there can be small variants, which are your single nucleotide polymorphisms, small insertion deletions, less than 10 to 15 base pairs, and also there were larger variations as well. So this project, which is the GTEx project, I'll just put the link here if you want to take a look. So here they describe the whole GTEx project. Just link here. Yeah. Sorry. Yeah. So now we had gotten the human genome sequence and variants that were present in the human genome, but we did not know the function of these human genome variants, right? So we knew that there are genes, these genes code for these proteins, but what happens if there is a particular variant in that gene? We, if it is particularly present inside the coding region of the gene, we know that probably it hampers gene expression somehow, or it hampers the production of the protein downstream somehow, but we do not know how much and by what extent, to what extent the variation would be seen, right? So the GTEx portal was launched around, I think, 2003, after the Human Genome Project was completed. So the major uh, targets of the GTEx portal, GTEx project basically was to get a list of variants that can affect gene expression, right? So, for example, I'll say if there is an intronic variant in a gene, we do not know if that, from a biological perspective, we do not know how that variant actually affects the gene expression, because it is not part of the protein coding region of the gene, but still somehow the variant is there, and we do not know how the variant actually is uh, sorry how the variant actually is contributing to the downstream processes. Is the variant actually important, or is it like just there and it does not contribute to anything, it's a synonymous variant? So that can also be possible. So what this particular program did was it established a comprehensive catalog of genetic variants that affect the gene expression across multiple tissues for research community to evaluate tissue-specific gene expression and regulation in many different tissues. So this again is a very important thing. So we know all the cells in our body have the same sequence of the genome, right? But still the functions of the cells are very different. So a liver function, liver cell gene expression will be very different for if you compare it to the gene expression of a lung cell or a neuron, like that, right? So but the genic region and the genome sequence is exactly same, but not all cell types will express all the genes present in the human genome, right? So the variants that are present in the gene, or the intergenic regions or the intronic regions, can affect the expression in different cell types in different manners, right? So that is also very important. So tissue specific expression is what they went for, because again, a variant present in the exact same location of the genome in liver cell versus the variant present in the lung cell, the impact of these variants are quite different. So it might be present in a gene that is not at all expressed in that particular cell type, so it does not actually hamper the gene expression of in that particular cell type, say for example liver cell, but it might be in the gene which actually codes for a particular protein that is used by that particular cell type to do some function. So that if a variant is present in that gene, that might actually impact that particular functioning of that particular cell type, right? So genetic variants that influence how genes behave are called expression quantitative trait loci. So this is a very important term. What we look for is basically, once we have found the variants in the gene, we want to know that if these variants actually contribute to change in gene expression in the cell, or is it like just present without any implications, right? So this GTEx data is used to enhance the functional interpretation of genome-wide association studies, findings from and identification of disease-relevant genes. So as we know that I think we have talked about GWAS a little bit in earlier classes. So basically, GWAS is a technique by which you identify variants present in a particular genome and try to find what traits are getting hampered by the presence or absence of that particular variant, right? So it's a linear regression kind of a thing. So on your y, so it's a simple y equal to mx plus c. On the y side, you have your phenotype value. Say for example, we are checking for a cholesterol parameter, say total cholesterol. So on your y vector will be your cholesterol values for, say for example, 10,000 people, and on the x vector will be the genotype information for this 10,000 people. And by genotype information, I mean presence or absence of a variant. And like, not only presence or absence, it's like it can be absent, or it can be present in a heterozygous form, or it can be present in a homozygous form. So we generally do additive models of genome wide association study. So the idea is that if you're, say for example, a variant is actually hampering hampering uh, the phenotype value of an individual person. Say for example, cholesterol, a particular variant is lowering cholesterol for a particular person. If that person is in a heterozygous form, the presence of that variant will reduce the cholesterol level. But if it is present in homozygous form, the level of the phenotype or cholesterol will be even lower. So that is what the additive model means. So generally, these kind of variants that we'll find in genome-wide association studies that are significantly associated with these traits are mostly intronic and intogenic variants. Very few genic region variants we find that are associated with these traits, because most variants that we have in our body are in the cell of in the genome are basically present in the non-coding regions of the genome. So if you, say for example, you do a GWAS and you find a particular variant, intronic variant, that is associated with a particular trait, you know that from a statistical standpoint, but from a biological perspective, it's hard to explain why that variant is actually contributing to the phenotype. So for that, information like expression quantitative trait loci become very important, right? So if you can study the individual that has that particular variant, if you can study their cell type and how the gene expression is getting varied because of that presence or absence of the variant, it gives you a biological understanding about how that variant is actually contributing to the phenotype increase or decrease, right? So yeah, so that was the whole idea of the GTEx project, that we find what actually these variants are contributing to, right? So they did this study and they created an online data resource, which is the GTEx portal, for storing, cataloging, searching, and sharing aggregated level data. And so a lot of papers have been published using data from the GTEx portal. Again, another important thing while checking for variants and how they are changing expression levels, gene expression levels in the cell, another important thing to consider is the ancestry of the person, right? So a person from African ancestry having a variant in the gene will have slightly different effect on the gene expression compared to say for example an Indian person having this exact same variant, right? So the 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 impact of the variant is also very ancestry specific. The reason being that the variant that is present might be tagged by other variants in linkage with that variant, right? So that linkage structure between different populations are different. So it's not the presence of just one variant. That one variant is not singly present. That will be present with some other set of variants, and it's kind of a system. It it's like, if one variant is present, another set of five variants will be present, and that set of five variants will be different for different ancestry people. So you are not actually looking at a single variant, you are looking at the whole structure, genomic structure around that region of the gene. So that differs from ancestry to ancestry. So the ancestry matched expression studies are important to find what actually is going on, right? So and yeah, GTEx data is integrated into genomic browsers like the UCSA Genome Browser and Ensemble. So you can visualize gene and variant information. And also there is a biobank they have developed for of tissue biospecimens of lung, brain, pancreas, skin, etc., as well as RNA, DNA, blood sample cell lines from around 1,000 donors. The GTEx Biobank also features an image library of tissue samples for researchers to browse the complete collection, and these are stored at the Broad Institute of Harvard and MIT, the same institute that actually came up with the GATK pipeline that we were discussing a couple of lectures earlier, right? So yeah, the idea here of the GTEx project is to find expression quantitative trait loci. What are these? These are basically variants that actually contribute to difference in or influence how genes behave and how the expression of the genes will change. So that was the goal of this particular project. Okay, so this is a schematic from the GTEx portal itself. So you have, say, multiple donors, a particular variant. First, the DNA will be sequenced and it is correlated with change in gene expression, and you'll find particular variants. So in this case, say for example, G2A is the variant, right? So this is actually not a variant site. Uh, the particular base is exactly same as the reference sequence. In this case, one of the bases have been changed G2A, and in this case both the bases have been changed from G2A, right? So this is basically a non-variant site, which is the genotype 00. This is a variant site with one copy of the variant present, which is 10, and this is both copies present in a homozygous condition, which is 11, right? So then they collect different tissue samples from the donors, brain, heart, liver, etc. They do genome sequencing, and then they collect these tissue samples, and then do RNA sequencing of across different tissues. So you can have separately for brain, heart, and liver, and then they check what is the gene expression in each tissue. Say for example, this is the liver tissue that they are checking. These are the variants that they are checking for. So GG, AG, and AA, and then they check what is of a particular gene, is the presence or absence of the variant is increasing or decreasing. In this case, you see, in the GG case, the expression of in the brain is very high. In the AG case, it is slightly lower, and in the AA case, it is much lower. So the presence of the A genotype here, or the A variant in this particular location, actually changes the gene expression in brain tissue, right? But you see heart and liver, it almost is the same. So again, that, as I was saying, these are like the gene expression is dependent on the tissue you are looking at, or the cell type you are looking at. So the variant might not be involved in changing gene expression in certain cell types. In this case, heart and liver, it does not change, but in brain, the gene expression is reduced by the presence of this variant, right? Excuse me. Now, after so this is what the GTEx project is all about. Now we talk a little bit more about EQTL. As I was talking about this, so it stands for expression quantitative trait locus. It is a specific region in the genome, or a locus, which contains a genetic variant like a SNP or a small indel, and is associated with the expression level changes of one or more genes. So it kind of is a switch that controls how much a gene is getting expressed or not. So that is the gene expression itself is a very complicated biological phenomenon, and that is controlled by a lot of factors inside the cell, right? There are some signaling aspects to it. There are some presence or absence of some substrates in the cell that contributes to it. There are some other genes that actually regulate the function of that particular gene. So it can happen like that. If you are having a variant in a gene that's function is, say, production of protein A, but what you're seeing is you do not have any change in the production of protein A with the presence of that variant. What you see is a decrease in the production of protein B due to presence of that variant. Although the gene that directly does not code for protein B, the gene actually controls the expression of the gene that actually codes for protein B. So it can be possible that the variant that is present in the gene, in that particular gene, that genic function might not be affected, but the function of it to control other gene expression, that might get affected as well. So it kind of is a control mechanism by which the gene expression is controlled inside the cell, along with other factors that control gene expression. So the major question here is, how do genetic differences between individuals lead to differences in gene activity, which is gene expression? And why is this important? Because it helps connect genetic variations to functional consequences, bridging the gap between genotype and phenotype. Because at the genomic level, when you have genomic data, you all do mapping, you do variant calling, you find the SNPs and all those things. You have the data that these are the variants that are present in the cell, in the genome, but you do not know what the function of that particular variant is. So that the concept of EQTL comes in. Right. So this is actually this is actually different types of EQTLs. So there are three types of EQTLs that are shown here. One is the static EQTL, another is the conditional, another is the dynamic, right? So yeah, the top part of the panel shows in which condition the EQTL influences a gene expression, or if it affects expression changes between cell types, right? So this is an EQTL, and there is a conversion of a cell type 1 to 2. How does it actually influence the this particular process inside the cell, right? And the B part actually shows so the first two actually shows how either the EQTL directly contributes to each of the cell type gene expressions, or it contributes to only one. So it is, first is the static one and second is the conditional one, right? The lower parts of the panel show exemplary mRNA expression profiles of the genes in 6 samples. The genotype of the EQTL in each sample is indicated by the color, assuming homozygous diallelic markers. A static EQTL impacts expression in all cell types. The ranking of gene expressions in per genotype is same in all conditions, as is the slope of expression change between cell types. A conditional EQTL, in this case, a conditional EQTL influences gene expression only in one of the two conditions, and thus gene expression is a function of genotype in one cell but not the other. So basically, the presence of the EQTL, where does the EQTL needs to be present to affect this particular process? Does it need to be present in both cell types? Does it need to be present in one cell type, or does it need to be present in some other cell not involved in this process, but that cell type is actually contributing to this particular process? So that can also be possible. Thus gene expression is a function of genotype in one cell but not in the other. The slope of expression changes may or may not be dependent on the genotype at the EQTL. A dynamic EQTL actually drives expression changes between cell types. This implies the slope of expression changes between the conditions are dependent on the genotype at the EQTL. So again, as I was saying, biologically, this EQTL can be contributing to a lot of things that is going on in the cell, directly or indirectly. Directly, it may contribute to the downstream protein production or other cell products, production of cell products in the cell, or it can indirectly affect something else that it is controlling. So that can be very interesting to study. So there comes the concept of two types of EQTLs, which are your cis and trans, right? So there are major differences between these two types of EQTL. So basically, the cis EQTL is located around 1 MB region along the gene, right? And actually they have to be present on the same chromosome. But in case of trans EQTL, it is located far from the regulated gene, either on a different part of the same chromosome or on a different chromosome entirely. That is also possible. So these become much more trickier to study, because a variant present in some other chromosome is actually contributing to the gene expression change in of a gene that is present in some other chromosome. So that biologically understanding that thing becomes very difficult. So next is the effect size. Effect size is basically the impact that particular variant has on the gene expression. So for example, if statistically speaking, if the effect size or the beta, which we talk about as the effect size, is say 2, what it means is the presence of the QTL drives the gene expression twice than earlier, right? So the effect size will determine how much the impact is, right? So generally C-sequentials which are present nearer to the gene they regulate, they are likely to have more larger and more direct and more easily detectable effects on the gene expression. And for trans-sequentials which are present far away from the gene, they tend to have smaller, more subtle effects that are more challenging to detect statistically. And in terms of mechanism, they are in C-sequentials, since they are present so nearby, they can directly affect transcription of the gene and through changes in regulatory elements like promoters and enhancers, right? So say for example you have a variant in the promoter region of a particular gene. So when the DNA polymerase will come and bind to the promoter region, it may not be able to bind because of the presence of that variant, because these polymerase enzymes, they are very specific to the sequence that of the gene that they bind to, right? So if you have a variant in that location, in and around that location, it is very hard for the enzyme to bind and do the replication or do the transcription. So it becomes very it becomes very impactful for the gene, right? So if the equatorial is present very near to the gene, it is much easier for it to regulate the gene expression. But if the gene and the variant are present far away, maybe sometimes on other chromosomes, it is harder to regulate, but they generally regulate the gene expression indirectly. And a common mechanism is by altering the expression of transcription factor or other regulatory molecules, which in turn affect the distant target gene. So it does not directly impact the expression. What it will do, it will impact the expression of some other gene, which the product of which is used by this particular gene to do its transcription, right? So that can be happening. So for example, I'll give you a good example like this. So there can be a variant in the RNA polymerase gene, which is the POLG or some genes like that, and those variants might not directly impact genic expressions of other genes, but since it is present in a gene which actually codes for a protein which is used by other genes to kind of generate mRNAs and proteins downstream. So presence of the variant in a gene like a polymerase is going to be very harmful and very impactful in terms of regulating gene expression for other genes as well. You do not know what kind of genes will be regulated because of a slight change in the polymerase activity. So the fourth thing is basically these are tissue specific. So they can be specific to certain tissues, but a significant portion are shared across multiple tissue types. And trans-equatorials, they are more likely to be tissue specific, reflecting their role in complex context dependent regulatory networks. And cis-equatorials are easily identifiable due to their strong effects and the limited search space for associations, because you are looking at around 1 MB regions in and around the gene. So it's much easier to find this kind of equatorials. But for trans, it is more difficult to detect due to their weaker effects and the vast number of potential associations that need to be tested across the entire genome. So it is again, so for cis-equatorial, it is very close by to the gene and it can affect the gene expression directly. But if it's a trans-equatorial, it is far away from the gene and it indirectly affects the gene expression. And it is much harder to detect in terms of finding equatorials. Trans-equatorials are much harder to detect than cis-equatorials. Okay. Moving on. So this is, excuse me. Sorry. Yeah. So I will talk about another project, which is the ENCODE project, which is actually kind of deals with this kind of equatorials only, but it is basically a international effort aimed at identifying all functional elements within the human genome, and it was launched around 2003, and its goal was to create comprehensive parts list of our DNA, going far and beyond just the protein coding genes identified by the Human Genome Project. So again, from the basic biology that we understand that there are protein coding genes in the genome, there are non-protein coding genes as well. So people used to think of the non-protein coding regions of the genome, which are your intronic regions, intergenic regions, and other non-coding regions of the genome. Those are actually junk DNA that we do not need them, right? But after the Human Genome Project, this project was launched to uh, kind of say that like only 1.5 to 2% of our DNA is coding for proteins. What is the function of the remaining 98%? We used to refer them as junk DNA because we do not know how this how it kind of how the rest of the this 98% of the junk DNA kind of behaves, or what is its purpose, right? So ENCODE's most famous and controversial finding was basically in 2012 they announced in a paper that the 80% of the human genome has some form of biochemical function. So this was controversial because uh, it's it was done using some statistical techniques and some validations, wet lab validations, but still biochemical claiming biochemical function was very difficult, but they still claimed it. So that was something that is there. But again, the main thing was that the myth was busted that we do not require intronic regions of the genome, we do not require uh, intergenic and other non-coding regions of the genome. That is completely not true, because they have some effects on the gene expression, right? So key discoveries included kind of enhancers, promoters, and silencers. So these are elements of the DNA which are not part of the gene, which are present much higher upstream of the gene, but these elements in the gene actually uh, correspond to functional expression of the gene. So presence of an enhancer will enhance the expression of the gene. The promoter is the place where the RNA transcription starts, and there are silencers which basically uh, reduce the expression of the gene. So all these elements are present in non-coding regions of the genome, previously thought to be as junk DNA. But this particular project, actually they understood the so-called junk DNA regions of the genome, and they kind of found these types of elements. These were regulatory elements that act as switches or dimmer controls for genes, often located far away from the gene they regulate. And the second finding was that the project found that at least 75% of the human genome is transcribed into RNA at some point in various cell types. This showed that cell reads far more of the genome than just protein coding genes. This was also biologically very interesting, because previously we thought only the genes that were transcribed were protein coding, but we found that there are a lot of genes in the human body that do not code for any protein. There are a lot of genes in a lot of genomic regions in the human cell, that human genome, that do not code for proteins, but they are transcribed into RNAs, but they do not ultimately code for protein, because there can be a lot of non-coding genes which actually code for different types of RNA. So for example, a gene has to code for a tRNA. The tRNA does not code for a protein, but it is important in in translation of the mRNA to proteins, right? So these kind of elements are also generated by these type of junk DNA sequences inside the human genome. And after that, they kind of did some regulatory network analysis, and they found that the gene expression regulation is incredibly complex, and the single gene might be controlled by a dozens of regulatory elements, and one regulatory element might influence multiple genes, creating an intricate web of interactions. So this was again reinstated. This fact was reinstated that gene expression is incredibly complex, right? So you cannot say just the presence of this variant, you cannot predict will actually lower or higher up the gene expression of that particular gene, because it is much complicated, because there can you may mutate one promoter, but there a gene can have multiple promoters that are there. You can cut off knock out a particular gene from the genome, but there will be other genes which are doing similar work or producing similar proteins to kind of take over the function of the gene that you have deleted. So again, it's a very complicated affair, and this project kind of helped us to understand that. Right? So moving on to some more recent projects, which is again the TCGA is a very well utilized and well known project. So this is the Cancer Genome Atlas program. So no, this is not the portal. Yeah. So this contains all the information for TCGA. So it is a genome research project which kind of only targets certain cancer types. So we sequence genomes for individuals that have cancers and also controls, so that you can compare. One second. Yeah. So the data is stored. The project is the Cancer Genome Atlas, but the data is stored in this portal called GDC, which is Genomics Data Commons Portal, which contains all the information. So this has around 86 projects, 69 primary sites. Sites is basically the site of cancer. So many cases, these many files, these many genes that they have studied, and these many mutations that they have found associated with different different types of cancer. Yeah, so you can see on the right hand side there's a plot given of how many cases that they have found for each type of cancer. So these are all given in thousands. So basically they have found around 8,000 bone marrow cases, 8,300, around 4,000 cases for breast cancer, head and neck, kidney, lung. So different types of cancer samples they have accumulated, and they've studied the genes, the genetic variations associated with these cancer studies. And so yeah, a lot of the data is under controlled access, so you have to apply for access and then get approved and then get the data mostly. But in some cases, say for example, let me type breast cancer. So these are so these are different projects, right? So let's take TCGA BRCA. So they studied the BRCA gene, which you know is an important part of breast cancer. So they studied this gene, and these are your different types of data available. So on the right hand side, you see you have an ETAC-seq for around 1,000 cases. Not 1,000. So you have ETAC-seq for around 74, but the total number of cases is 1,000. You have diagnostic slides, so they do kind of biopsy reports and those kind of things. Genotyping arrays. So basically you genotype the SNPs that are present. You do methylation status, you do miRNA-seq. So these are much more advanced things that they do. You do basic RNA-seq, which is the gene expression thing. You do whole genome sequencing, and WXS is basically, it can be whole you can do whole genome, or you can just do the exome regions of the genome targeted sequencing as well. So you have a lot of information in terms of the the types of data they have generated. And here uh, uh, s- so these are the different experimental setups, and these are the different types of data that they have. They've copied CNV data, which is very important in terms of cancer, because CNVs are basically copy number variants. So basically, a particular copy of the genome, a stretch of the genome, present multiple copies or deleted off completely. So if it is present in multiple copies, it is a duplication event. If it is deleted completely, it's a deletion event. So in case of duplication, the copy number is higher. In case of deletion, the copy number is lower. So you have a lot of, basically all the type of information that you can think of. You have biospecimen, clinical, CNV data, DNA methylation data, proteomic data, sequencing data, SNV, small SNV data, so somatic structural variation data. So again, in cancer, what happens is basically, if you are um, uh, if you are in contact with some carcinogen, you will get cancer. Say for example, UV light can cause skin cancer, right? But you are not born with those variants. Your germ cells or your other cells in the body do not possess those variants. If a certain types of cell actually come in contact with those particular carcinogens, it will mutate the DNA of that particular cell only, and you'll generate cancer from that particular cell. But it is not something that you're born with, right? So for that, you have to do somatic variant analysis. When we talk about variant calling, we generally talk about germline variant, the variants that are present in all cells of your body. But in case of cancer, it is only present in a small set of cell types that is causing the cancer, right? So those are somatic variants compared to germline variants. So we need to study somatic variations again. So they have all these particular information, and uh, these are your case IDs, and again, so many information. Let me see if I can find anything else. uh, no, so this is let me see what information they have for the biospecimen. Yeah. Yeah, so they have all the sample. These are your sample IDs. These are your different case IDs. These are different information, how the samples have been processed, what kind of data files they have been generated, what preservation method they have used, what is the sample type that they are looking for. So it can be a primary tumor, it can be some derived tumor, it can be some metastatic tumors, and so on. And what kind of samples that they have taken. Have they taken solid tissue? Have they taken blood? Etc. etc. So these all sample information is present in this particular file. In other things, you'll find slide information as well. So how if there are malignant cells present, proliferating cells, infiltration cells, and so on, lymphocyte infiltration, and necrosis, and so on and so forth. So these data are very like this particular type of data is easily available. What is not easily available is the sequence data. So if you want to find this particular copy number variation data or your sequencing rates data, you have to kind of apply for access, and they'll approve your access, and then only you'll get the data. Otherwise, it is not possible. So this is the TCGA portal, and it's one of the most widely used databases for genomic research in terms of cancer. This is the widely used. So we are having again. Let me show you a smaller thing again. So we are kind of trying to build the Indian version uh, of the TCGA database, which is known as the ICGA, which is the Indian Cancer Genome Atlas, looking through for Indian genomes, that Indian individuals that have cancer. We are collecting those data, and sorry. So currently they are looking at large scale multi-omics projects for profiling of breast cancers. So this is the Indian version of the of TCGA. This is ICGA. So this is very recently launched, I think a couple of years back. So yeah, good. Okay. So again, so in TCG you have over 33 different tumor types, 10 rare cancers, and it has collected and systematically analyzed over 20,000 primary cancer and matched normal samples. So matched normal samples meaning like you have the same individual. Say for example, the person has skin cancer. You take cells from the cancerous portion of the skin, and you also take cells as control from the non-cancerous or the healthy portion of the skin to all compare, right? So those are matched normal samples, which are basically the samples from the same individuals of cell types that do not have cancer. Okay, so yeah, as I was talking about, so you have a lot of information, a lot of data present in these in the data portal. You have WUS data, which is just the exome sequencing. So difference between whole genome and whole exome is basically in whole genome you sequence all 3 billion base pairs in the human genome. In whole exome you only sequence the exonic regions of the genome, the region of the gene that actually codes for a protein. So those are, that is targeted sequencing, which is the whole exome sequencing. Now also on top of that you have CNV data, RNA sequencing data for transcription profiling. For epigenomic profiling, we saw that there are DNA methylation data, and there is also proteomic analysis as well. So this is a comprehensive multi-omics project which actually provides data of all kinds that you can think of. Right. So for that we have for accessing TCGA data. Sorry, for accessing TCGA data we have this data platform, which is the GDC. This was the page that I was showing you. So you see this is actually the GDC portal. This is not the TCGA portal. So this is a GDC portal which actually contains the information, the data from TCGA project, right? So the GDC portal is basically this is a data commons portal established in 2016, and it kind of takes all the data and puts it in its database, and it can give you raw genomic data generated over many years at different centers, right? So the GDC reprocessed raw TCGA data. So what it did was it collected data from all sources, and then it reprocessed the data using standardized state-of-the-art bioinformatics pipeline. So they took the raw data and they aligned it to a particular same build of reference genome, same build of other bioinformatic pipelines that it were using, so that there is no batch effect among the data. So the sequencing data is the only raw data that is present, and then they have done the processing on their own. They have not taken processed data from it everywhere, right? So now what they've done is they all DNA and RNA sequencing data they have realigned to GRCS38, which is kind of the latest. After this, the T2T has come out. So yeah, and then somatic mutations, they were recalled using standardized pipelines, which is the Mutec2 is the most commonly used software for this, and it improves the accuracy and consistency of mutation detection across cancer types. And TCGA is itself one of the largest projects within the GTC, which also hosts other major cancer genomics datasets. And yeah, so you have every data file from raw sequencing reads, which are BAM files, to processed mutation calls, which are your variant called files. They're assigned this particular unique identifier, and which creates a permanent traceable record of the data's origin and processing history. So yeah, this is the TCGA poster. So they have about 2.5 petabytes of data. That's a lot of data. So again, to put into perspective, 1 petabyte of data is equal to around 212,000 DVDs. So that's a lot of data. It has 33 tumor types, 10 rare cancer types, 11,000 patients, 7 different data types, including genomics, transcriptomics, proteomics, uh, your biospecimen samples, doing biopsy reports, and so on and so forth. And yeah, so right now they have around 20 collaborating institutes, but anyone can deposit their data in the TCGA portal, so you can do that. And this is the way uh, the GDC portal has worked. So what they do is they take the raw data, they convert it into FastQ, they do alignment, and they do pre-alignment QC, quality control, and data processing in a standardized pipeline across all raw data, so that the data that you are getting, even the processed data, there should not be much variation in terms of batch effect on those kind of things. There should not be variations in terms of the data that is getting out from that portal. So yeah, so that is all that I have in terms of discussion. Now we'll move on to the assignment sections. If you have any questions. Today's class is mostly like describing this different data portals and all, but yeah, do not think that these are not useful. These are very useful things. These are very widely used, and a lot of researchers use this to publish their papers, and we have a lot of interesting work going on because of the availability of these kind of data sets. So getting data is very important, and specifically for bioinformatics people like me. So yeah, let's go on to the questions. If you have any questions to ask. Okay, we'll move on to the questions. Okay, I'll give another minute. this is simple. We discussed about these two projects, ENCODE and GTEx. Uh, since we are talking about eQTLs and uh, GWAS associations observed in non-genomic regions, so these kind of projects used RNA sequencing or expression data to kind of validate the findings that if a particular variant, even if it is not present in the genic regions, it can affect gene expression. So these two projects are the answer: GTEx and ENCODE. Moving on. Okay. Yeah. So option B and C are correct. So DNA-seq is basically a technique used. They use an enzyme called DNase, which actually cuts open regions of open chromatin regions. So these tell you if it is able to cut, then there is no histones present there, so it is a more accessible region, and if it is not able to cut, it is a non-accessible region. Then you have ETAC-seq, which is very widely used, and it uses a transposase to simultaneously cut and tag these open chromatin regions. And yeah, it's both of them are very widely used. So Okay. Yeah, that is correct. Bisulfite sequencing is very widely used in terms of DNA methylation and getting information in terms of methylation status. Yeah, that's true. Moving on. Okay. Yeah, that is correct. 1 and 3 are correct. So E-clip is basically enhanced cross-linking and immunoprecipitation. So it is an immunoprecipitation assay which checks the binding of RNA with other proteins. RNA bind and seq, which is basically RNA binding by next generation sequencing. It basically is designed to determine the sequence specificity of some RNA binding proteins. So yeah, it is also an RNA binding assay. So these two are correct. The other two are actually, those are also RNA analysis. So cage is used for gene expression analysis, and rampage is used for annotation and mapping of promoters. So those are fine, but those do not use binding assays. So yeah. Okay, so the idea is that we already knew about protein coding genes, right? So genomic elements in the protein coding genes were already studied before the sequencing and also projects like GTEx and ENCODE. But after that, the rest of the type of genes, small RNA, uh, LNs, long non-coding RNAs, pseudogenes, the annotations of these have become much more simpler, and understanding functional impact of these genes have become much more substantial after the sequencing of the human genome. So, other than the protein coding gene, the rest of the three are the answers basically. So, yeah. Yeah, except for discovery of new species, you can do biomarkers, you can do association studies, which are your GWAS, you can do novel basic biology, projects like ENCODE and GTEx, and so on. So, yeah, that's correct. Okay, the answer is biobanks. So basically, what we do is when you collect phenotypes and so for this kind of study you need to collect phenotypes and you also need to collect genome samples. So basically blood samples they collect, they uh, sequence the genome from the blood cells, and they use the phenotype values to do genome wide association studies, right? uh, Along with that they kind of store these biospecimen samples in biobanks. So in large biobanks you'll see that. So for example, the UK biobank is a very well-known resource for that. So uh, they have around 500,000. Sorry, is there a question? Okay, let me just do this first. So UK biobank is a very good resource. So they have this they have 500,000 people data. So all sequencing data they have, all types of biobank data they have. They kind of yeah. So let me tell you the UKP 400K. This is the recent paper from the UK Biobank where they have analyzed these many individuals. That's a huge number. That's the largest that we have till date, and they have done a lot. They have done sequencing. They have done association studies. So yeah, they have done associations. They have done sequencing. They have done biobanking of these samples, and they have done imputation and so on and so forth. They have done a lot of things. So this is the 500K paper, but they have done it on 150,000. They have done it on initially I think 100,000 and a little less than that. So these are very important biobanks. So they store the data. They store the data. They also store the biobank samples in a biobank. So uh, uh, biobanking is a important thing in terms of association studies. Now let's go back. PGSA, I think, let me check once, I think if I'm correct. Uh, I'm not very sure about PGSA. I don't know if it's actually a thing. PGSA. Hmm, I don't think that's a correct. Uh, I don't think that's anything like it's a wrong thing that is given there. So to ignore it. I don't know what that is. Yeah. Okay, the answers are these two, that functions cannot be maintained indefinitely without selection, and if 80% of the genome would be functional, all of that would have been conserved. So it is, both of them are from an evolutionary perspective. So if 80% of the genome is functional, then we should have 80% similarity with other species. So, for example, chimpanzees or something like that, because the functions are not going away, right? So, even if the species are changing, the closest ancestors should have around 80% similarity, around 80% conservation, right? More than 80% conservation. So, the conservation part is the first one, and second is without selection these functions cannot be maintained indefinitely. So the function has to be a good or a bad function, right? So it has to be selected, either it has to be positively selected or it has to be negatively selected. So the best logic that kind of counteracts that 80% of the genome has some function is basically from an evolutionary perspective, not from a phenotypic expression or a gene expression perspective. It is the perspective that if it is 80% is functional, that there should have been selection across the 80% of the genome, but we only see selection in genic regions, we only see selection in exonic regions, mostly exonic regions and those kind of things. So that kind of contradicts the logic. So both are from an evolutionary perspective, that there has to be some selection if it has so many functions, because we do not know what those functions, and like if the selection has happened or not. Definitely the selection has not happened, because functions is not the direct function, right? The 80% of the human genome has some function does not say that it has direct function. It is not coding for anything, but it is helping or regulating something else, right? So that is the truth, but it's not truly functional. It is functional, but it is not functional in a sense that if you do not, if you, it does not directly code for anything, or it does not directly do anything. It regulates via intermediaries. So both from a evolutionary perspective, it's correct, right? So these two options will be the correct ones. Just a second. Okay, so basically, which options did you say? A and C. AG19 and GRC is 37. Basically, AG19 and GRC is 37 are the same thing, and AG38 and GRC is 38 are the same thing. So it's basically all four. the answer is all four. AG19 is an acronym for GSE 37, and EG38 is an acronym for GSE 38. So it's just like it is different, written in a different way, but AG19 and 37 are same, AG38 and GSE 38 are same. So, and it has done using both the versions. So yeah. Okay. It's actually all. No, I think this is a wrong answer. The ataxic is a technology, so this is not a project. So yeah, first three should be correct. The fourth option is I don't think it is given correctly. Ataxic is not a project, so yeah, ABC is correct. Correct. Yeah. Yeah. Option B is correct because the other ones do not exist. So yeah, GTEX is the correct option. Yeah. I think this is a single option, correct? So it's C. High C is there. High C does the like configuration technique, but CS high C, I'm not sure if it is. It's C high C. It's, so it's CS high C. High C chromatin. Yeah, it's high C. CS high C, I'm not sure if it is. Yeah, it's called just high C. But high C is a correct option. We use high C data for uh, chromatin structure. Yep. Yeah, that's correct. The rest of them are actually sites in the genome. CT-safe is the only protein. So this is basically, it does cross-linking and immunoprecipitation. So yeah, sorry, CTCF, it binds to DNA sequences within the insulator elements, and you can help identify these things. Yep. Yes. Yeah, the uh, genome arc is not there. I'm not very sure, guys, about non-human genome projects, so I need to check as well. Genome arc. Okay, I think GenomeArk and the Vertebrate Genome Project are the same thing. So main reference will be stored in the GenomeArk. So I think GenomeArk is the storage, like just like TCG and CDC. I think GenomeArk is the storage area and VGP is the project. I do not have much knowledge about these things, so sorry for that. Yeah. Yeah, so basically, I think GenomeArk and VGP are the same. VGP is the project, it is the data portal. Okay. Yeah, I think the answer is four, if I'm not wrong. Yeah, this is the fourth one. I do not have much information about these thing, guys. So forgive me for this. So yeah, the fourth option is correct. I'm not sure about the exact working of this. Oh, okay. It's good. So I also don't know the answer to this, so let me check. Mineralized teeth in bird. Oh, this looks like paper. Mm. Single loss of mineralized teeth in common action. Single loss, yeah. Lost in a single event. Okay, that's great. Okay, moving on. Yeah. Okay. Yeah, this would be option C because EQTL, if it is having higher log odds for a segment, that means EQTL of that segment is associated with that phenotype. So that is the whole thing of EQTL mapping, right? So if that particular element or the particular variant in the genome is actually contributing to the phenotype or not. So yeah, it means higher association if it is higher log odds. What higher log odds mean is basically presence um, of that particular variant or that region increases the phenotype value. So it is positively associated. So that is what it means. think this question had some issues. Uh, okay, option A. I also think it is option A, but I think this question had some issues. My answer is also option A, but yeah, none of these I'm not sure why it is given like that. But yeah, the idea is that if the variant is present in and around the gene, it is a cis-equatorial. Trans-equatorial, it will happen if the variant is present in some other chromosome or some very downstream or something like that, very far away from the gene. So my answer is also cis-equatorial. I'm not sure about why it is given none of this. I'll uh, raise this with sir, and I'll get back. I don't think this answer is correct here. So yeah. Yeah, so the developmental equatorial does not consider adults. So it considers infant, prepubertal. So after birth till puberty is just after puberty, before becoming an adult is the phase. So you do not consider an adult for the developmental stages, right? So yeah, without the adult, the rest of the answers are correct. So this is quite simple. So yeah, first let me tell you the difference between EQTL and SQTL. EQTL, E stands for expression quantitative trait loci. S stands for splicing, right? So, uh, uh, as the name suggests, if it is an EQTL, it regulates expression, and if it's an SQTL, it regulates splicing. So in this case, if it is favoring one particular isoform over some other gene, it relates to splicing. So splicing will give rise to different isoforms. So since it is not given what type of if the SNP is far away or is nearby, it can be both trans SQTL or cis SQTL, depending on the location of the SNP. But if it is favoring one particular isoform over others, it's basically favoring one splicing form over other. So it relates to the splicing. It's similar to EQTL. It's just it is involved in the splicing mechanism, not in the expression mechanism. So yeah, I hope that is clear. Yeah. Yeah, so it'll be both D and B because the SNP location is not given. It can be far away, so it can be a trans SQTL or it can be a cis SQTL. So, yeah, so that's all from my side. If you have any questions, you may ask. Sorry, my camera is off. Human. So this is the T2T. This is the paper. This is the complete sequence of the human genome. This came out in 2022 from the T2T consortium. The T2T consortium is the telomere to telomere consortium, which basically means like one into another sequencing they did. So right now we have dissolved the telomere regions, the mostly centromeric regions. Most of the regions of the genome are sequenced and resolved. Y chromosome has also been resolved using the T2T consortium and some other regions as well. So human is the answer. I'm not sure about the other organisms, by the way. I just told you human because that is the most recent one. If this is a single option correct, it's definitely human. So this is the T2T consortium. Yeah. There are some good videos explaining this if you want to take a look. Sorry, Y chromosome is shortening the telomere. What does this mean? I did not understand the last sentence that you wrote. In general, Y chromosome gets shortened, like that's through evolutionary mechanisms. The Y chromosomes get shorter and shorter. I'm not sure if it is shortening the telomere or not. Okay, like that. Yeah. In general, the Y chromosome gets shorter and shorter. All the chromosomes get shorter and shorter with age, but with evolution, the human chromosome, Y chromosome, has gotten shorter, much shorter than what it was supposed to be, and it is getting shorter by the day. But Y chromosome does not have a telomere per se because it's a very unique chromosome present in the human genome because only the males have it, that too as one copy. So it is very hard to classify uh, uh, in that say, but kind of it is shortening. So it is a very tricky region of the genome. So it is very hard to quantify the telomere. There are telomeres in the Y chromosome, but it is very hard to quantify which region it is, because we did not know the sequence of the Y chromosome till very recently. So yeah. So there are also haplogroups in Y, which basically are distinct signatures of ancestry of the human race. And there has been a lot of studies on the Y chromosome that has linked us to African early out of Africa events in terms of population genomics and so on and so forth. Yeah. But in general, with age, the consistent pattern is that the telomeres get shortened. So with this, a lot of diseases also gets used to it. So there is a lot of neurodegenerative diseases, Parkinson's and Alzheimer's. There are a lot of diseases like this which are associated with the shortening of telomeres of certain chromosomes. And with age, this happens. That is a natural process of the telomere getting shortened. Okay, then if no more questions, I'll close the session. Okay. Okay, before I leave, next week, Tuesday is the uh, s- last class. Wednesday, I'll be taking a kind of a recap session of the whole course, same time, 2 hours. I'll be recapping the whole course Wednesday, I think 17th. 16th will be week 8, and 17th will be the whole course recap. Uh, Exam wise, I have got no clue. I do not have that information. You have to ask the NPTEL people, or bring this up in discussion forum. I do not have that information, so I can't help you like that. But yeah, so next Tuesday is the 8th week, and next Wednesday I'm taking an extra session of 2 hours, which will kind of, I'll cover from beginning to end as a kind of recap before your exam. 20 September for what? Oh, okay. I'm not sure about that. But yeah, 17th September is the extra session that I'll be taking. So I think you'll get a mail regarding that. Okay. Thank you. Thank you for joining. I'll close this session. We'll meet next week. Thank you.